All right, ladies and gentlemen, we're going to try and finish from what we did not do today. This was a slide that we ended with. I want to make sure that we are straight on this. You know, as light, we've already talked about how light comes through the lens, gets flipped. All right, it hits these rods and cones in the fovea and the retina, or not necessarily the fovea, but definitely in the retina. To which case a chemical reaction, the photo transduction occurs, which is again the changing from a mechanical energy or stimuli to a neural or stimuli, and that's what happens here. Light hits it, hits the rods and cones. Rods and cones then talk to bipolar cells, which then activate the ganglion cells. All right, these we didn't really mention those much. They're sort of go-betweens between the sensors and how the information gets sent. Bipolar cells activate the ganglion cells, the axons of which converge to form the optic nerve. So all these axons from all these ganglion cells all sort of come together and they form the optic nerve. And that's what transmits the information from the visual cortex via the thalamus and the brain. And here, when we were talking about the blind spot, here's another example of that. No, I'm sorry. This is a rod. I'm sorry. No, I'm wrong. This is not the blind spot. This is a rod-free area. So you've got rods and cones, rods and cones, rods and cones, but when in the fovea, you don't have any rods. And rods hold on. Yeah. We've not talked about what rods and cones do yet. All right, but I believe, if I remember correctly, the cones are responsible for color, and rods are responsible for your ability to see in low light situations. So, if this is the fovea right here, all right, and it does not have any rods, which mean low light sensors, your eyes will see the most clear, obviously, when there's a lot of light. All right, and we're going to watch a video tomorrow that kind of talks a little bit about the difference in rods and cones. But rods focus or deal with being able to fire in low light situations, whereas cones, think color, cones fire as they respond to color. So this next little piece here, you'll probably see this tomorrow or the next day in maybe a bell work or maybe a quiz. This is essentially what it says, how the eye sees. All right, so, and you may get this exact thing, I haven't decided yet, but how does the eye see? You know, this is sort of the whole process from beginning to end. Light waves pass through the cornea, which is the eye protection. That's the shiny part. All right, and then the pupil, which is the adjustable opening, and the lens. Now, obviously, you don't have the iris here, which is the muscle, which is the adjustable opening. So maybe you could probably put iris under adjustable opening, but just to make sure you know what it is. So the next part. Okay, there's the iris. The iris, the colored muscle, controls the amount of light entering the eye by controlling the size of the pupil. The lens changes shape. It's not like glasses where it's the rigid. The actual lens in your eyes can change shape and accommodates to focus the incoming light onto the retina. As the light strikes the retina, the light energy activates the rods and cones, which are your photoreceptors. The central area where the rods and cones cluster is the fovea. I'm going to have to adjust this before I post it online. Signals from the rods and cones and collected by the bipolar cells transmit the information to the ganglion cells. The ganglion cells' axons are bundled together to form the optic nerve, which transmits information to the thalamus and then the visual cortex in the brain, which is located in the occipital lobe. The optic nerve leaves the eye, creating a blind spot because no receptor cells are there. All right, so you'll probably see something like this in the next day or so. So we're going to talk a little bit about the optic nerve, blind spot, and the fovea. The optic nerve, again, carries neural impulses from the eye to the brain. The blind spot is a point where the optic nerve leaves the eye, because there are no receptor cells there. This creates a blind spot. We will see this tomorrow. And then the fovea, again, is a central point on the retina around which the eye's cones cluster, meaning that there's a lot more of them in that area. All right, now photoreceptors. We have not quite talked about them much. I'm going to talk about them a little more tomorrow. But we're talking about rods and cones. The cones, there are 6 million cones, 120 million rods. Their location in the retina, they're kind of central in the cones, but in the periphery of the retina of the rods. The cones do not function well in low light, but the rods do function well. The cones are color sensitive and detail sensitive, where the rods are not. And again, bipolar cells and ganglion, I've mentioned them other than just passing, but the bipolar cells receive messages from the rods and cones and take them to the ganglion cells, all right, which sort of bring everything together to form the optic nerve. And here's another example of that. Light enters here, hits the fovea. This is pretty much the exact same thing we just saw earlier. Just to review, hits the rods and cones, which then create neural impulses, which is gathered up by the bipolars and then sent to the ganglion cells, whose axons will create the optic nerve, where you have a blind spot right here. Cones, as I mentioned earlier, they're important in seeing color and in seeing detail. Whereas rods are sensitive in dim light. They're the things that make you first aware of an object's presence, but they only respond to black, white, and gray. Nocturnal animals have a lot more rods than cones. Raccoons, bats. Well, bats don't see well, but lemurs or whatever. All right, if you are an animal that spends most of its time awake at night, you don't really need cones because you don't need to see color. You just need to be able to see in dim light. So they They are mostly fire, and your brain was going to interpret that as black, white, or gray. Visual information processing. The optic nerve connects the thalamus in the middle of the brain, and the thalamus connects to the visual cortex. If you're going to think about the order, think of just of it in alphabetical order. Optic, thalamus, visual, OTV. The one back here, talking about just reasons to give you all to kind of help remember the order. Think PBG or PBJ. Bipolar ganglion. So as you are receiving the information in, it is processed. Optic nerve, remember, this can be confusing here because your eye will bring in the sights from both the left and the right side of your eye. And the ganglion cells are going to be from the left and right side of your eye. They're going to come together around the thalamus. But what happens here is strange. The visual cues from the right side of both eyes end up here, and the visual cues from the left side of both eyes end up here, and then are processed in the visual cortex. So study this a little bit to where you sort of understand. We talk about the left side of the brain focuses or, controls the right side of the body. Well, in your eyes, it's a little strange. It's a little different. Both sides of the right eye are controlled or are processed in the visual area thalamus on the right, and then the left side of both eyes goes through the visual area on the left side, also in the thalamus, and then they come together again here in the visual cortex and the occipital lobe. All right, feature detection. Nerve cells in the visual cortex respond to specific features such as edges, angles, and movements. And as you get older, you sort of hone this in. I can remember as a kid, my dad, my papa, and my uncles being able to see deer in the woods just randomly. We'd be driving down the road, and, oh, there's a deer, and 3 or 4 of them say, oh, yeah, there it is. But as a kid, I'm looking, I don't see him, all right? Now I know the reason why I couldn't see him is because I did not have those feature detection. I didn't know how to deal with the feature detectors or what those were. I didn't know how to interpret those. Now, driving along the road or walking along in the woods, I can see the feature detections of a deer. And by that I mean usually the flat back or the upright ears or something like that. You may not be able to see the whole thing, but I can see enough of it for my brain to realize, oh, hey, that's a deer. All right, a couple experiments on vision. Hubel and Weisel's experiment. There were some cells in the visual cortex respond only to certain types of visual information. For example, a diagonal line moving up and down. All right, so they just tested these cells in the visual cortex, and sometimes they wouldn't fire until certain feature detectors were present. Like I said, a line like this, or even a flat line. All right, only then will those certain cells fire. All right, and shape detection occurs in different areas of the brain. And again, they've done this through just experimentations. They'll map. Will hook electrodes to the scalp, and that will measure electronic activity, and then they'll just show people things, and different parts of the brain fire on different things. And this is what they've mapped. This is just a small portion of what they mapped, but there's 2 parts of the brain that fire when we're thinking about faces. There's another part of the brain that fires when we see a house, when we see chairs, when we see houses and chairs. So there are different parts of the brain that fire when you're dealing with different types of features. And now it brings us to perception in the brain. Our perceptions are a combination of sensory, bottom up, and cognitive processes, top down. All right, and here's an example of bottom up and top down. All right, all we have are a bunch of lines, but we sort of put these lines together. Whereas this, what do we see here? Do we see 3 Pac-Men, or do we see a triangle? Parallel processing is fairly important. This processing of several aspects of the stimuli simultaneously is called parallel processing. The brain divides a visual scene into subdivisions such as color, depth, form, and movement. So when we see something moving, there are 4 different things going along. So when I see a deer bounding through the woods, first thing I see is there's 4 things happening there. I see the color of the deer, the brown and the white. You see the motion, and we've already seen what happens or what can happen when somebody suffers from motion blindness. You just see it, and then all of a sudden it's over here, and then all of a sudden it's over there. But you see that motion. Then you see the form. I see the actual outline of the deer. And then I also am processing depth, how far away it is or how thick it is. All these things are happening at the same time, and that processing is known as parallel processing. And here are some examples of this. When parallel processing fails, and a lot of times you see this in stroke victims. Mrs. M may not be able to perceive movement where people will suddenly appear and then disappear. And then pouring liquid into cups again seems frozen because they don't have that movement. All right, you can't see the movement of the liquid. And then blind sight, when you can, somebody will put something in front of their eyes and then move it. People will say, well, I never saw it. Well, they may not have seen it because they couldn't perceive the fact it was moving and then gone. But when asked a detail about the object, they get it right because their brain was able to process it. So and that's why it's called parallel. It doesn't all happen. 4 things are all happening at once. So even if you lose one, all right, you'll still be able to perceive the other things, whether you realize it or not. So, and here, this is a nice little graph kind of focusing on how we go from sensation to actually recognition. Remember, the last part of perceiving is our conscious perception of us actually consciously experiencing it. So you start with a scene. In this case, it's looks like 2 high divers. The retinal processing happens, the actual physical part, the receptor rods and cones, bipolar cells and ganglion cells. The light sort of goes through that. Then we kind of goes in our brain, hits the feature detectors. Those brain detector cells respond to specific features, edges, lines, and angles. So we actually see that we recognize these are divers just based on the angle and the fact that, and then the perception or depth perception happens in the next one with parallel processing. Brain cell teams process combined information, so everything goes into parallel processing. Then you see color, movement, form, and depth. And then finally, recognition. The brain takes these things and constructs the image information based from stored images. So we our brain relates those things too. So this is sort of the 4 major parts or 5 major parts of us seeing something, anything. And then we kind of get to a question that Louise asked, about why is my shirt black? Do objects possess color? We'll see. Is a lemon actually yellow? Is your shirt actually black? Is chili pepper actually red? No, light has absolutely no color. Let's talk about it for a second. There are different theories as to why we see color. All right, the first one I'm going to talk about is the trichromatic theory. And this kind of gets back to the cones, and you're going to learn a little bit more about this in the video we're going to watch tomorrow. But the human eye has only 3 types of cone receptors that are sensitive to different wavelengths of light. You've got the short, which we talked about, the blues. We hadn't really mentioned medium, but that's sort of the greens and yellows. And then your long wavelengths are your reds and oranges. People see colors because the eye does its own color mixing by varying ratio of cone neural activities. So when we see, let's say, this color, all right, it's definitely not violet, but it's definitely not red. What is actually happening is that the photoreceptors in our eyes, the different cones, are firing in different combinations. So there are different combinations of this versus different combinations of this, right? Or different combinations of this. So I see this color based on the amount of cones firing that deal with blue, red, and green. Just kind of like throwing a bunch of food coloring together. Those are just firing off, and whatever combination is happening is where I'm perceiving this blue color or this green color. The other, so that is the trichromatic theory, and it deals with the three types of. Now, this is not theory. The fact that there are three types of cones. All right, I don't believe. I think this is pretty much proven with science. But how we perceive that, I think, is what the theory is. Now, this is a different theory. This is opponent process theory. And this was from a guy named Herring. And he suggested that we process 4 primary colors combined in pairs of red and green, blue and yellow, and black and white. So we've got the 4 primary colors here, plus black and white. And here's how they went through that. Take a look at this and stare at the dot in the middle of the flag. About 30 seconds. All right, so this is red. This is green and yellow. So stare at it. You can pause it. I'm going to move on, but you pause it for 30 seconds. And then as I leave it, or as I move, you should be able to see red and blue and whites. And here's the reason why that is. Look here. When you see blue has the opposite of green, or blue will connect to green. This chart doesn't make sense. But the opposite color is the one you see. So here, the green would be red. I'm going the wrong way. The yellow would be blue. Yellow and blue. And the white would be black. So the British flag is, the stripes are all green. I'm sorry, the stripes are all, I can't even get it right. The stripes are all red. The flag itself is blue, and these parts here are white. Well, when we stare at this, we see green, black, and yellow. But when we move on, then it changes. So there's the theory is that we process these at the same time. And finally, we get to color blindness. This is a genetic disorder, which there's usually 2 or 3 in every class that is colorblind or suggests they're colorblind, which people are blind to green or red colors. This supports the trichromatic theory. Guys are a lot of times more colorblind than girls. Here are a couple examples. This is from the Ishihara test. There are 2 different numbers in each one of these bubbles. I believe this one should say 74. And this one should be 45. And take note if you do not see those or if you see something else. So, and that's all we're going to talk about with vision today. Maybe it wasn't as long as I thought it was going to be. Maybe it was. But we will go ahead and do some things tomorrow in class. Being able to do this kind of allowed me to do some things in class I wasn't going to be able to do previously, but now I can. So hope you all had a good night, and we will talk to you all later.